Hello everyone, welcome to the third session of Genetic Introduction. So we are discussing about the three main terminologies which are usually confused in genetics, which are a chromatin fiber, the chromatin and the chromosomes. For knowing these terms in detail, first we'll see what is a cell cycle, because these particular terminologies are usually referred to this particular event which is called as cell cycle. So we'll see what is a cell cycle, what happens in this, etc. And after this discussion, we'll move on to what are these structures and when are these structures seen in the cell cycle and what are their structural specialities, etc. So coming on to the cell cycle, as you see in the diagram, this is a cyclical event where a particular cell grows and divides. So this cycle continues. That means a particular parent cell starts its life, it grows, it divides into daughter cell, and each daughter cell again moves to this cycle. Those cells it grows and then it goes for division and moves on. So such a cyclical event that happens for every cell in our body is called as a cell cycle. So this particular cyclical event is divided into 4 phases depending upon the events that happen in each phase. And the 4 phases are starting from, or the starting phase of a cell cycle is referred to as the G0 phase, which you see starting from this right-handed side. Okay, so we'll start the discussion. So this is the first stage, which is called as, it starts from here. The life of a cell, usually we talk about a cell cycle. It starts from the phase which is called as G1 phase. It is also referred to as gap 1 phase, also called as growth 1 phase. That is, the G represents both gap phase or a growth phase. And this is followed by the second phase, which is called as S phase, followed by the third. This S phase is nothing but, S stands for synthesis phase, followed by the growth 2 phase or gap 2 phase, which is followed by division, which is M phase. That means, as I said earlier, a particular cell, it starts with a nucleus. It starts at the G1 phase. It grows, it produces its important protein, etc. It grows, it moves around this particular cycle. It reaches the M phase, which is the dividing phase. Mitosis or meiosis happens in the M phase, where a single cell, if it is mitosis, it divides into 2 daughter cells, and these 2 daughter cells has the same fate as that of the mother cell. That means it also follows G0 S, that is G1, not G0, G1 phase, S phase, G2 phase, which is followed by M phase. Each of them again divides, and those daughter cells also the same, follow the same fate. And this cyclical event that happens is usually referred to as the cell cycle. And if you take the particular 4 events in the cell cycle, division happens only in the M phase, and in the rest of the 3 phases, which is a G1, S and G2 phase, the cell is growing, or growth of the cell takes place during this 3 stages, which is G1, S, G2. Because it is divided into 3 distinct phases, it is to be known that different types of growth occurs in different phases, which we'll talk about, or we'll discuss it in the coming parts. So the G1, S and G2, which is the phases where the cell grows, is also collectively called as the interphase. So interphase is a collective terminology for G1, S and G2 phase, and it is in these 3 phases the cell is growing. Or you can also term it as, it is the phases where the cell is preparing itself for the phase which is called as division. Preparing in the sense that this particular cell is growing and depositing the proteins, extra amounts of all the organelles, etc. And this is being done by the cell is for the next coming event after interphase, which is called as M phase, where a single cell is going to divide into two, and this particular single cell has to deposit everything which is to be shared to the two cells, and that is what is meant by growth that is happening for individual cells. So this particular cyclical event that is happening in the cells of our body is called as the cell cycle. This happens in eukaryotic cells, which is having a nucleus, etc., mitosis, meiosis, etc., happens. So this cell cycle is usually connected with the growth and dividing cycles of a eukaryotic cell. So coming to another phase, which you see here, which is G0 or gap zero phase. So that is the phase where the cell is not dividing. So you might be knowing that there are some cells in our body which stops growth after a certain period of time. For example, our muscle cells and the neurons. After a neuron is completely being formed, after a neuronal cell is differentiated into a neuron, it does not divide. It stays as neuron throughout the life. It might degenerate, but it does not divide. That means it is not taking part in any events of the cell cycle. That means during embryogenesis, that means during the event of neuron formation, so these the neuronal forming cells, they are differentiating from the cell mass of the embryo. They get differentiated, they move to different cycles, they divide, they produce daughter cells, etc. And after a particular round of cell cycle, it get differentiated into a so-called neuronal structure with the dendrites, with the axon, etc. After such a structure is formed from the neuronal precursor cell, so every cell in your body is having a precursor cell from which it is formed, which is a cell of the embryo or a single celled embryo. So from that non-differentiated cell, it passes through several cell cycle, and it is now differentiating into the cell which is called as a full-fledged neuron. After a full-fledged neuron is formed, it is not taking part in the event of cell cycle. That does not mean that that cell is not alive. The cell is live. It does the normal metabolic functions. It produces its protein, it produces the neurotransmitters, it functions. But the only difference is that that particular cell is not taking part in the events of cell cycle. That means it does not move on to an S phase, it does not move on to G2, it does not move to S phase, it does not divide. A neuron or a muscle cell, the same is applicable to the muscle cell, the skeletal muscle cell. After a skeletal muscle cell is formed, it is not dividing. So those particular cells, after they get differentiated, it is not getting, or it is not taking part in the events of cell cycle. Such cells, they are moved into a position which is outside the cell cycle, because they are not taking part in the cell cycle, and that particular stage of that cells is referred to as the G0 phase. So mind you, G0 is not a phase where the cell is dead. The cell is completely alive. The only difference with the cells which is taking part in cell cycle is that they do not undergo any events of the cell cycle. That means they do not divide. They do not produce extra proteins which is required to be compartmentalized into two different cells. They are alive, they are doing all their normal metabolic functions, they function as a neuron, they produce neurotransmitters, they carry out impulses, everything, they are functioning normally. The difference is that they are not engaging in the events of cell cycle. So this is the event which is called as a G0 phase. Coming to the interphase, as we said, now we'll discuss what are or what happens for a cell in different phases of the cell cycle. So in different phases of the cell cycle, as you can see in the diagram which is towards the right hand side, in the G1 phase, which is that this particular area, which is the G1 phase. In the G1 phase, the growth and normal metabolic roles of the cell is being done. So it is at this G1 phase, the particular cell which is engaged in a particular event which is called as this cell cycle. In the G1 phase, that particular cell synthesizes the maximum protein, that means the highest peak of protein synthesis happens during this G1 phase. After G1 phase, there is no protein synthesis. So no protein synthesis. So wherever we talk about no protein synthesis, you should keep in mind that that particular DNA is now in a condensed state which is not suitable for transcription. Is that clear? So only in the G1 phase your DNA is compatible for protein synthesis. After that, your DNA is going to be compacted because it is going to divide. After G1 phase, the DNA will be becoming more and more compact than the solenoidal structure, moving to the structure which is called as chromosome. So the maximum of all the proteins that is required for the two daughter cells, the enzymes, the structural proteins, the membrane proteins, whatever is needed, whatever protein is needed for both the cells will be synthesized. And it also requires proteins for the next event, that means S phase, because in each phase proteins are required as enzymes, etc. So those proteins which are required for S phase and those proteins which are required for G2 phase, those proteins which are required, because in M phase you might have studied the division where the chromatids or the or everything inside the cell will be divided into two daughter cells or compartmentalized into two daughter cells. So the events where the spindle fibers forms, etc. So spindle fibers are proteins. So all the proteins of the spindle fibers, etc., everything is being formed during the G1 phase. After G1 phase, there is no synthesis of proteins. So synthesis, or the event of protein synthesis, so the synthesis of more and more protein is usually referred to as growth of the cell. Increase in protein content of the cell is usually referred to as growth in a cell. So that happens in the G1 phase. So that is where the cell is synthesizing and storing more amount of proteins which is required for the different events of the cell cycle, and also required to be transferred to the daughter cells. So that was G1 phase and the events that is happening in a G1 phase, which is followed by the next event which is called as S phase, or the synthesis phase. And this is the phase where DNA replication occurs. DNA replication in the sense that when a single cell produces two daughter cell, DNA should be there to be given or to be segregated into the second daughter cell also. So there will be an increase in the DNA content or doubling of the DNA content during this particular stage of the cell cycle, which is called as S phase, where the DNA content or the nucleic acid content doubles. The content, the DNA strand or the DNA content doubles during the process of S phase, and it is for distributing, or it is to be segregated into two different cells during mitosis. So that is what is called as S phase, followed by G2 phase. G2 phase is also called as growth 2 phase, but growth is not similar to G1, and this is a very short phase. G1 is a very long period in a cell cycle because the cell is synthesizing the major amount of protein, etc. So that takes a bit more time than the other one, which is called as the G2 phase, which is shorter phase. Synthesis of minor protein occurs because DNA is already going compacted. So some events of protein synthesis at some genes occurs during the G2 phase. In some cases, no protein synthesis occurs. And usually it is said that the G2 phase or the growth 2 phase is usually referred to as a preparatory phase, where the cell is preparing for division, which is the M phase, where no synthesis of protein, etc. happens. The cell is preparing itself for division. Preparation is nothing but the coiling of DNA to form a structure which is called as chromosome. So the process of coiling or compaction, etc., that is the event that usually happens in the G2 phase of the cell cycle. G2 is followed by M phase. M phase can be mitosis in somatic cells and it can be meiosis in gametic cells. So whatever the events may be, a single parent cell which has completed G2 phase or which is eligible for carrying out M phase, it will divide, producing the daughter cells. If it is mitosis, it will produce the daughter cells which is similar to the parent. If it is meiosis, it will produce the daughter cells which is having half the number of chromosomes as that of the parents. So these are the events in the cell cycle. And what is, so in this particular diagram you can see the different events, that is you can see prophase and metaphase and anaphase and telophase, which are the different events in the mitotic phase of the or if mitosis is happening, the M phase can be split into that 4 different phases. So this is all about what is called as a general introduction of what is called as a cell cycle and the different events in the cell cycle. So here you can see this is marked where the G1, S and G2 combined together, it is called as the interphase. So now we'll see. Okay, so now we are going to see how our DNA is behaving throughout the cell cycle. Behaving in the sense, what are the different structures that is attained by the DNA during the events of the cell cycle. So now we'll see the different structures. So we'll start with the structure which is the normal double helical DNA. So we'll start with the double helical DNA. So as we said earlier, in a normal cell, this particular DNA is seen in the form of a solenoid. So in the G1 phase, if I am taking, so this is not the cell, I am taking the nucleus. If this is the nucleus, inside the nucleus in the G1 phase, you can see the DNA in the form of a solenoid. So when a solenoid is formed, a single DNA will form a single thread of or a single rod of solenoid. A single or a double or a double helical DNA will form a single fiber of solenoid. It can produce, or when it you can just imagine you have a double helical DNA and you are compacting it, that will produce a single fiber of solenoid. So in such a single fiber in the G1 phase is referred to as a single chromosome. So that is referred to as a single chromosome. You can take it, or you can take the term chromosome in this particular situation also to know what is a chromosome. As I said in the first session, chromosome is a terminology that is usually to be restricted to a structure that is seen in the metaphase stage. But usually you use this terminology elsewhere. We use it to make a connection between the structure of the particular terminology of chromosome and the structure. So that is being used elsewhere. That's why me to use this particular term chromosome here. So here you have a single, if you are having a single this particular double helical DNA, and if it is getting compacted, it will form a single solenoid. And in this particular G1 phase, uh, if this is a human cell, you can see such 23 pairs of solenoidal threads. That means inside this nucleus, if it is a human cell or a human nucleus, if you observe the chromosomes, or if you are counting the number of solenoidal threads inside the nucleus of a human cell during the G1 phase, you can see such threads with number called as 46. You can see such 46 threads of solenoids. Okay, and if this particular solenoidal thread is getting compacted, is going to compact, so this compaction usually occurs only during the mitosis or the prophase, etc., or when it is moving to the cell cycle. But imagine this particular single, or if this is a single solenoidal, so this is a single DNA strand. A single in the sense it is a double helical strand which is now compacted into a solenoidal fiber. And this particular DNA strand, if it is going to be compacted, or it is moving to be more and more compacted, and if this structure we neglect everything, that means synthesis etc does not happen for this. I am taking this particular solenoidal thread directly into the M phase. This is just imaginary. This is for getting clarity into what the confusing terms are, which are chromatin, then the chromosomes, and the other chromatin fiber, etc. So here, if a particular solenoidal thread, imagine it is getting compacted in the G1 phase itself. It is getting compacted. If it compacts to form a chromosome, the structure will be like this. Okay, it will attain a structure like this. And such a structure, which is attained by the compaction of a solenoid, is usually referred to as a chromatid. Usually, as I said earlier, the term chromatid only applies to a complete chromosome that is usually seen during the metaphase stage. But here, to understand what is a chromatid, or how, or what are the structural details of a chromatid, we are just hypothesizing the situation. That is, if you have a single or this particular DNA is a single chromosome. For example, this is chromosome number 1 of a human, the first chromosome. A single chromosome means it is a when you consider a chromosome, mind you, you should remember that it is a double helical DNA, a stretch of double helical DNA. And when it gets compacted, it forms a fiber, a solenoidal like fiber. And if we are compacting that particular fiber more and more, you get a structure like this, which will have a depression in somewhere in the middle. So this depression, usually you draw it in the middle, and such depression is usually referred to as the centromere. So this depression in every case need not be in the middle. So there are other instances, or there are other peculiarities for the centromere. We'll talk about it when we discuss with mitosis, meiosis, etc. So this particular, we are talking about a chromatid. If a solenoid like this is getting compacted, imagine it is getting compacted, and if it gets compacted, you get a structure like this, and this particular structure is called as a chromatid. It is a single chromatid. And if you are untying this chromatid, what can you see is you get a double helical DNA in this. So a double helical DNA which is compacted. So when you talk about a chromatid, imagine that, or it should come to your mind that it is a single, or it is a single stretch of double helical DNA. Is this clear? So this is what a chromatid means. So now we were talking about, so such structures can be formed if you are compacting a chromatid, or if you are compacting the solid 46 chromatids. This will be the structure which is present in the G1 phase. That means if you assume the formation of a chromatid from the DNA or from the chromosomes which are present in the G1 phase, you can number it as, if it is a human cell, and if chromatids are formed during the G1 phase, the number of chromatids that can be formed from the chromosomes from a human cell during the G1 phase is 46. You have 23 pair. So just imagine 23 pair means it is 46. So each of the 46 is a solenoidal fiber. As we said, it is a single stretch of double helical DNA, and that is going to be compacted. So when that is compacted, you get such a structure. So such 23 pairs are there, and if you count the number of chromatid, it will be same as 23 pair or 46. Hope this is clear. And now we are moving on to the next stage, which is called as the S phase. How this chromatid. So from now we are going to consider this as our chromatid, and we are going to see how this chromatid is behaving in the cell cycle. Throughout the cell cycle, how changes happens in this particular structure. Okay. So we said inside the cell or inside the nucleus, now we are having such structures which are single chromatids. So if it is a human cell, in the G1 phase, you will have 46 such chromatids. And now the cell is moving on to the next phase, which is called as S phase or synthesis phase. So in synthesis phase, we said that your DNA will double, or the DNA content will double. So there are some terminologies which are confusing, or there are some concepts which are confusing here, and these concepts are usually taken to produce or to build up question for many of your entrance examinations, etc. With this particular difference or confusion in terminologies. So I hope the first term is clear. What is a chromatid? So now we are going to see what happens for this chromatid in the S phase. So in the S phase, the event that happens is nothing but DNA replication. That means we are having a single, or a double helical DNA. This double helical DNA will replicate. It will produce a daughter DNA, which is same as which will be an exact copy of the parent. If this is the parent one, it will produce a daughter DNA which is exactly like a copy or a clone, because it is copying. So DNA replication is nothing but copying of one strand of DNA to other, copy and paste. So what is being present in the parent? The exact thing. If A is present in the, if an alphabet A is present in the parent at one position, the same A will be copied in the same position for the daughter DNA strand. So and if this is the daughter DNA strand or the daughter DNA which is being produced from this, so this is particular, so this is what happens. So this is the event. So this is the event that will happen after during replication. So during replication, from one strand you get two strands of DNA. Is this clear? So usually these two strands which are formed, they will be closely opposed to each other. The one which is being produced from the other DNA strand, they will be usually closely placed to each other. And this, if this particular DNA, this was the DNA is the DNA. This is a structure of a single chromosome after it is being replicated. So now we will see if this is going to be compacted into a structure like this, what will be its structure. So it will be like this. So this was the first structure, and now it has created a copy of this, and as I said, it will be connected at certain points, or this will be the structure. So how many chromatids are there in this particular structure? So I have drawn this arrow. It is nothing but I am compacting this into a solenoid and then into a chromatid. So this particular structure we saw in the previous discussion, such a structure will be producing one chromatid like this, and now in the S phase it has replicated or created a copy of itself by DNA replication. And after DNA replication, you can see that there are two double-stranded DNA strands, and that two DNA double-stranded structures, which is copy of the other, if they are getting compacted to form a chromatin like structure, you will get a structure like this. I will draw it more perfectly. They will be joined. You might have seen the uh, diagrams for that. So if this is a double helical DNA and now this has replicated, and if this is compacted, you will get a structure more or less like this, which is usually seen in the books, the structure of a chromosome like this. So it is after S phase you can see structures like this. That means the DNA has, so from here DNA has replicated and the structure will be like this. So in this particular event or this particular time, if you count the number of chromatids, so in this case we saw 46 chromosome and the number was 46 chromatid. In this particular phase of S phase, if you count the number of chromatid and the number of chromosome inside the human cell at S phase, you can see that the chromatid numbers are double. That means there were 46 solenoids. Each 46 produced its copy, and that copy is now clipped with one another. The parent as well as the daughter will be seen opposed to each other like this, and this will be counted as chromosome number one, because one is a copy of the other. You cannot count it as a second chromosome. It is not a different chromosome, it is the same chromosome itself, because it is the copy of the first one. As a result, you can see, or if you take the number of chromosome, the chromosome number will remain the same. There will be 46 chromosome itself, and each chromosome or each solenoid, as we talked earlier, or each chromatid which was in the G1 phase is now replicated. It produces one more chromatid. As a result, the chromosome number is remaining the same, 46, but the chromatid number doubles, because each chromatid of the G1 phase has produced its own copy. So now one chromatid for the first chromosome is now into, or you can count it as 2. The other can be counted as again 1 into 2, etc. So every 46 chromosome or the 23 pairs has now replicated and produced its own copy of chromatid. That means the number of chromatids doubles, but the number of chromosomes remains the same. That means in this particular stage, a human cell will have 46 number of chromatids and 46 number of chromosomes and 92 chromatids, because each one chromatid like this has now produce its own copy. So now it is 2 for one chromosome. Like this, now you have 20 or 46 chromosome which is having this particular structure. As a result, the number of chromatids will be doubled to 92. The same, because no change happens in G2. In G2 also the same number of chromosomes and chromatids can be seen, which is 46 and 46 chromosome and 92 number of 46 and 92 number of chromatids. The same will be visible at the start of meiosis. At the start of meiosis, at prophase and metaphase. In anaphase the situation is different, which we'll talk about it in the next session. So till metaphase stage, you will have, for a human chromosome or a human cell, you will have 46 chromosomes and 92 chromatids. So this is how this particular structures are being or structures behave during the cell cycle, or these are the different. So we have talked about the chromatids. What are chromatids? I think that's clear. And we talked about chromosomes. So chromosomes, they are nothing but the utmost condensed structures of the DNA that is seen only during the metaphase stage, but we use the term misappropriately at many locations. The exact term is given for the structure that is seen only during the metaphase stage. And coming to chromatin fiber is nothing but the solenoidal structure that is seen, or that we talk about during mitosis and meiosis. So during the cell cycle, if we are taking the solenoidal structure, or we are talking about a solenoid, you don't call them solenoid, we call it as the chromatin fiber. If you see your olden textbooks of mitosis and meiosis, you can see that chromatin fiber condenses to produce chromosome. So that chromatin fiber is nothing but the solenoidal structure. But when you talk about a solenoidal structure in cell cycle or during the events of cell cycle, you call them as chromatin fiber. So that is with the three terms. Now coming to two other terms, which is sister chromatids and non-sister chromatids. So sister chromatid means this structure that we saw here. So these two are called as sister chromatid. That means it is derived, the DNA strands which are derived or which are copy of each other, which are derived by replication of one of the DNA strand. So such two DNA strands are referred to as, so such DNA strands or such chromatids are usually referred to as sister chromatids. So these two are sister chromatids. These are the sister chromatids. That means the DNA strands which are produced as a result of replication of one of the strands. That means they two are clones or exact copy of each other. That means one looking at the mirror image of the other. That is, they are same or ditto of one another, and these two are usually referred to as sister chromatids. And coming to the other terminology, which is non-sister chromatid. So if you are comparing the chromatids of different chromosomes, so this is of the same chromosome. If you are comparing, or if you are taking another chromosome here, and if you are comparing these two, they are usually referred to as non-sister chromatids. That means these two sister chromat or these two chromatids, they are not copy of each other. So sister chromatids means they are copy of each other. That means they have the same copy of genes, same type of genes, etc. At because they can be considered as photocopy of one another or clones of one another. And non-sister chromatids, we talk about non-sister chromatids when you talk about another terminology we are coming on to, which are homologous chromosomes. So when you talk about human chromosomes, we say that we are having 23 pairs. So this 23 pairs of chromosomes is nothing but they are called as 23 pairs of homologous chromosomes. So 23 pairs means so we are grouping this total 46 into 23. That means you have 23 chromosome, and each 23 is represented twice. Represented twice means you have two sets of 23 chromosomes. Two sets means one set, that means one set of 1 to 23. For say, I will erase these ones. Okay. In the case of humans, we are having chromosome numbers 1 to 23. 1 to 23, and that is seen in pairs. You have two sets like this. This makes the 46 chromosome. Of this, one set you get it from the mother, and the other set you get it from the father. So such a pair, that means you will have two first chromosome, you will have two second chromosome, you will have two third chromosomes, and such pairs of same chromosome. They are same chromosome, but they are not identical, because one you have got it from the father and the other you got it from the mother. They are similar. These two chromosomes, the homologous chromosome means they are chromosomes which you get one from the father and one from the mother. That means if I take, so if these are two homologous chromosomes, I take it as chromosome number 1. So this chromosome number, you get it from the father, and this you get it from the mother. So if you compare these two chromosomes, they will have the same genes at same position. That means if you're having the genes for hair color, you will have at this position, you will have the gene for hair color for this first chromosome at this position itself, because they are homologous or similar chromosome. So what is the difference? The difference will be, if this is a hair color, if mother is having the hair color, this is both are the gene for hair colors, that is a similarity. And what is the difference is that sometimes father will be having a brown pattern of hair color, and mother will be having a white pattern of hair color. So that is the difference, and this is what determines the hair color of the new or the young one. So this is or these two are now considered to be homologous pairs. So such 23 homologous pairs are present in a human, in every human cell, which makes up to 46 chromosome. So in this particular structure, we can talk about the sister chromatids and non-sister chromatids. So if you talk about a sister chromatid in this case, we'll erase this. Okay, so these two, with no doubt, we call them sister chromatids because they are copy of one another. They have replicated. So consider this particular structure. We are talking about at metaphase after S phase. They are replicated, and now these two, as I drew here, they are said to be sister chromatids because they are a copy of each other. And non-sister chromatids, these two can be called as these two are usually called as non-sister chromatids. These are called as non-sister chromatids because the features of non-sister chromatids are they are not derived from the same parents. As you can see in this, one is derived from the mother and the other from the father. That means they are part of homologous chromosome. The similarity between non-sister chromatids is that they will have same gene order. Gene order in the sense, as I said earlier, if you have a hair color gene at this position, the non-sister chromatid will also have the hair color gene at the same position. But the type of hair color will be different. In this particular case for sister chromatids, here also they will have the gene for hair color, but this will be the same, it will be brown. If this is white, this will also be white because they are produced by replication. And these are called as, or these two are usually referred to as non-sister chromatids. So non-sister chromatids usually will have the same genes, or they are the main thing is they are derived from different parents, and the similarity is that they will have same genes at same locations, but being different parents, their expression might be, the expression means what they produce might be different, what that genes produce might be different. They might be the same hair color, they might be the same skin color, but the pattern of skin color will be different. So that is the difference with sister chromatids and non-sister chromatids. We also talked about homologous chromosomes. So these two chromosomes can be called as homologous chromosomes. And the next is non-homologous chromosome. That means if you are comparing chromosome number 1, the two chromosome number 1s, you call them as homologous chromosome. And if you compare chromosome number 1 and chromosome number 2, which is entirely different, which will have entirely different genes, entirely different size, entirely different gene organization, etc. If you are comparing chromosome number 1 and any other chromosome, apart from its homologous pair, such two pairs or such two chromosomes are usually referred to as non-homologous chromosome. So now we have completed 4 terminologies: sister chromatid, non-sister chromatid, homologous chromosomes, and non-homologous chromosome. So these are a bit confusing. That's why the video is a bit long. I know, I have seen the time of the video, it's something around 45 minutes. I took more time to elaborate on each and everything. But there might be confusions again regarding the terminology. So please feel free to come up with the doubts, because we'll clear this. We'll study this thoroughly and move to our main topics. So expecting your doubts, clarifications, etc. With this, we come to uh, we are coming to the conclusion of the introductory part. Again, we have one more slide to be completed, something around 10 or 15 minutes, which will be dealt tomorrow. We'll deal with that tomorrow. And that is with the different genes and the structure of genes and how they are arranged in a DNA, etc. So we'll discuss about genes, paternal, maternal, etc. So those terminologies tomorrow. With this, we have completed the most confusing terminologies we have completed now. So just go through the videos. If there are doubts, please feel free to ask, because the questions which I have framed in the assignments for today is completely based on this particular video. So watch the video carefully, clear your doubts, and move on to answer the questions in the assignments. Thank you.